Okay, so last but not least, Dr. Elliott Robinson, who is tasked with the impossible mission to bring us to lunch within 10 minutes. Elliott going to talk to us about visual hypersensitivity in mice model of NF1.
Okay. Great. Thank you very much. This is my first RASopathy meeting. I'm just really happy to be here and to get to meet folks and interact with the patients and their family. It's wonderful. So I'm going to talk today a little bit about a project that has been led by Zoe Capel, a graduate student in my lab who is not here today, although I have her poster out there. So this is only 10 minutes. I could talk for 2 hours. So please come find me during lunch and ask any additional questions. And so what I want to talk to a little bit today about is sensory hypersensitivity that we've noticed in animal models of RASopathy syndromes. And so the ability to detect rapidly approaching objects and have a motivated behavioral response faster than the actual conscious perception of that stimulus is one that's highly evolutionarily conserved across species, whether you're talking about mice or primates or even humans. And we need systems in our brain to detect these rapidly approaching threats to avoid harm that can come as a result of them. Right? Because if you don't have these systems in the brain, then that you can experience real physical harm. And in rodents, and because my lab studies animal models, and so in rodents, they are really attuned not to rapidly approaching objects from their level, but from above. Right? These are nocturnal animals that forage and must avoid predators that hunt visually from above. And so in rodents, we can model the innate defensive response to a visual threat, such as a bird from above, through something called the looming stimulus assay. So a looming disk basically starts as a small black dot that appears on a screen above and rapidly expands. And this simulates the shadow of a bird coming down on the animal. And so when a mouse is in this arena here and a looming disk appears, it causes rapid flight to shelter followed by long periods of freezing. And so why should we as the RASopathy community care about this innate defensive response from a rodent? Well, we should, because one thing that we've noticed and we discovered completely by chance studying an entire other system of the brain is that mouse models of RASopathies originally in mice modeling neurofibromatosis type 1 are more sensitive to looming visual threats. They have an increased probability of escape that when you present them with a disc and their escape velocity is higher. This is something that I originally found working at Caltech as a postdoc in Viviana Gradnaru's lab that collaborated with Marcus Meister's lab who determined or who developed the looming stimulus assay that we've replicated now in the lab. And so we use this apparatus here in which we have an IR backlight, we have an infrared camera, and so the mice are completely in the dark except for a screen above the threat zone. And over here on the right, there's a little shelter that they can escape through. It looks clear to you, but it looks opaque to the animal because again, we're using IR to basically see right through it. And we can track the animal's motion. So we not only know if they escape, but we know exactly when they're escaping, how fast their velocity is, how complex their path is, etc. And this system was built by Austin Fisher in our lab. And so when we started the lab, we wanted to know, is this a bona fide RASopathy phenotype? So we got a mouse model that was developed by Dr. Ron Wacklaw at our institution at Cincinnati Children's in collaboration with Carlos Prado as he was there. And actually, Cassidy Grover in my lab is going to speak later today about the development of and validation of this model. But it's a mouse model of MAPK1 related RASopathy, in which if you look at the MAP kinase pathway here, the mouse has a gain of function mutation in ERK2, which is the terminal MAP kinase. And so we wanted to know that would a activating mutation in a downstream MAP kinase recapitulate a phenotype we saw in an NF1 model mouse in which they have loss of one copy of the NF1 gene acting at the level of RAS. And yes, it does. So we've shown that now that if you have activating MAPK activating mutation downstream in the pathway, you produce a mouse that is more likely to escape in response to a visual threat, and that they have higher escape velocity and they show show more freezing when tested later on. And so again, why do we care about this? So one, we think this models some aspects of autism spectrum disorder, right? When you have increased sensitivity to salient or arousing sensory stimuli. We think that this is important to kids who maybe have issue controlling their emotional responses to things that are arousing to them. And also ADHD, again, where distractibility can cause by the inability to suppress responses to things in the environment, which makes it difficult to focus. And the other reason why we like the looming stimulus assay is that the neural circuits that mediate these responses are very well described. And it gives us a system where we can go node by node within neural circuits and say, like, if you drive MAP kinase signaling in this part of the circuit, does it reproduce the phenotype? And this is important because we think by doing these experiments and understanding these behaviors, we might be able to learn something bigger about how the brain works in RASopathies. And so that by studying the responses to looming visual threats, we may understand like something greater about cognition. And so that this is like a big question of our lab that Zoe sat out to look after. So there's a number of circuits involved in the response to looming visual threats. One, computations regarding the detection of looming threats begins in the retina, in the eye. And these retinal ganglion cells that are the main output neurons of the eye that make up the optic nerve are important for the response to looming visual threats, where it's been shown that these off-transient alpha retinal ganglion cells are needed for the response to looming visual threats. And so we've actually partnered with Greg Schwartz at Northwestern and the Feinberg School of Medicine, who's one of the premier retinal electrophysiologists. And we know that in mice modeling NF1 that have loss of one functional copy of NF1, there is absolutely no change in the sensitivity of the retina to light, either their spontaneous firing rates or in their response to like a light stimulus across multiple types of retinal ganglion cells. So we don't think it's the retina projection to the eye. And here's the data, just a small bit of Greg's work looking at these off-transient retinal ganglion cells. Okay, so the ability to detect the threat and then make a decision about whether to escape evolves the midbrain. And so this has really been looked at really nicely by the work of Tiago Branco, which has shown that the perioccipital gray area and the superior colliculus are necessary. So the retina projects directly to the superior colliculus, which through projections to other midbrain sites like the perioccipital gray or the ventral tegmental area is necessary for escape decisions and producing the behavioral response. Additionally, we know that dopamine release, dopamine, a neurotransmitter we think is important as cognition and being affected in ADHD, is really important to the response to looming visual threats. So my lab now has spent a lot of time looking at this in a few papers that we published in the lab, and we now know that dopamine release in the nucleus accumbens is needed for the behavioral response to a looming visual threat, and that if you just use a laser to pause dopamine release exactly when that visual stimulus is presented, the mice will not escape at all. And this is important because years ago as a postdoc, I showed that dopamine release evoked by a visual stimulus was enhanced in mouse models of NF1. And so finally, we know that the cerebral cortex, which is an evolutionarily newer part of the brain that's more developed in humans and primates, that is really important to learning and memory and things like that, has top-down control over all these subcortical centers. And so if you have a mouse without a cortex at all, that they still have responses to a looming visual threat. So you don't need a visual cortex to perceive and have an escape response, although the response becomes dysregulated, where they have repeated responses and never habituate to the stimulus. And so in order to ask the question of how does MAP kinase in different parts of these circuits, like how can it produce the phenotype, we used genetic mouse lines where we could drive MAP kinase signaling specifically in different neural progenitor lineages, so during development in different parts of the brain, and say, does this enhance responses to looming visual threats? And so we use these Cre recombinase lines where in only the cells that express this Cre protein could you then have expression of an activated MEK1 mutant to drive ERK signaling immediately upstream of ERK. And such as in the developing midbrain here, where the superior colliculus and the PAG are that we know are important for escape decision, as well as in the developing cortex and the EMX1 lineage, which gives rise to all excitatory neurons, astrocytes and oligodendrocytes in the cortex and hippocampus. as well as inhibitory interneurons, which we know are important for learning and memory deficits in NF1, but also the striatum, which is important for motivation. And the take-home message here is that only MAPK activation in the developing cortex produces enhanced responses to looming visual threats, whereas it produces a loss of function in the other brain regions, which again was much to our surprise. We thought the midbrain was the answer, and it turns out it was the cortex. And so our hypothesis is that, at least in this case, that there is some loss of top-down inhibitory control from the cortex that's enhancing responses, behavior responses to these arousing sensory stimuli, that somehow loss of the ability to inhibit these urges or these innate responses is causing these phenotypes in these RASopathy animal models. And now working with Ron Waclaw in our transgenic mouse cohort, Cincinnati Children's, we have developed a new animal that will allow us to express these MAPK1 mutations only in the cell types of interest, so we can determine, is it the excitatory neurons in the cortex that drive this, is it the white matter, or is it the astrocytes? And with that, I can take questions. Thank you to funding sources, the NIH, the Simons Foundation, the Gilbert Family Foundation, the DoD, and others. Cassidy is going to talk about that MAPK1 mouse model today, but please come find Avery at her poster. She has all these nice bracelets for the meeting, and we'll talk about gene therapies.
Thank you so much.